Have you ever wondered how organisms build their complex structures from simple building blocks? It's a question that has fascinated scientists for ages, leading to the field of biosynthetic studies. This fascinating area of research seeks to understand how life, in all its complexity, emerges from fundamental, simple components. Biosynthetic studies are the detective work of biology. As detectives, scientists have to piece together the clues to understand the intricate processes that happen inside the cells of organisms. They examine how simple molecules transform into complex structures, how cells grow and divide, how tissues form, and how all these processes are finally coordinated. These studies are crucial to our understanding of life itself. They help us comprehend how organisms grow and develop, how they adapt to their environments, and how they maintain their health. They also shed light on what can go wrong, leading to disease and decay. The knowledge gained from biosynthetic studies can be applied in many fields, from medicine and agriculture to environmental conservation and bioengineering. Now you might be wondering, how do scientists go about studying these intricate processes? Well, they use a variety of methods, each with its own strengths and limitations. Some of these methods include tracer techniques, which allow scientists to track the movement of molecules within an organism. They also use studies of isolated organs, tissues, and cells to understand the specific processes that occur within these structures. In addition, scientists use grafts and mutant strains. Grafts involve combining tissues from different organisms to study their interactions, while mutant strains allow scientists to understand the role of specific genes in biosynthetic processes. Each of these methods provides a unique perspective, helping to build a comprehensive understanding of biosynthesis. But as with any field of study, biosynthetic studies are always evolving. New techniques and approaches are constantly being developed, pushing the boundaries of our understanding. The journey of discovery is never ending, and each new finding opens up a world of new questions. So, are you ready to explore this fascinating world of biosynthetic studies? Let's delve deeper to understand how scientists unravel these intricate processes. Imagine being able to trace the path of a single molecule through an organism's metabolic pathways. That's what tracer techniques make possible. Yes, this is not a page from a science fiction novel, but a reality in the world of biosynthetic studies. Let's delve deeper into this fascinating world. Tracer techniques are a potent tool used in biosynthetic studies to track the journey of molecules within an organism's metabolic pathways. Now you may wonder, how does this happen? Well, the answer lies within the concept of labeled molecules. These are essentially molecules that have been tagged or marked with a detectable substance, allowing scientists to trace their path through the organism. Imagine a group of tourists navigating an unfamiliar city with GPS trackers on them. The trackers allow their guide to monitor their route, identifying where they go, what they interact with, and where they end up. Similarly, in biosynthetic studies, these labeled molecules act like those GPS trackers, illuminating the complex maze of metabolic pathways. The beauty of tracer techniques is that they allow us to follow a specific substance through an organism's metabolic pathways. We can track the substance from the moment it enters the organism through its various transformations and interactions, all the way till it's excreted or used up. This gives us invaluable insights into the workings of life at a molecular level. But that's not all. Tracer techniques have wide-ranging applications. They help us understand how drugs interact with the body, how pollutants affect ecosystems, and even how our bodies process the food we eat. They've been instrumental in medical research, environmental science, and even agriculture. In a nutshell, tracer techniques are like a high-tech detective, tracking the journey of molecules, revealing their interactions, and uncovering the secrets of metabolic pathways. They are an essential tool in the quest to understand the intricate workings of life, from the smallest cell to the most complex ecosystem. Tracer techniques illuminate the invisible paths of life's building blocks. So, the next time you gaze upon a living organism, remember there's a whole unseen world of metabolic pathways illuminated by the brilliance of tracer techniques. Sometimes to understand the whole, we need to study its parts in isolation. This statement rings particularly true in biosynthetic studies where the power of reductionism shines brightly. Reductionism in biosynthetic studies is like using a magnifying glass to examine the intricate details of a beautiful painting. It's all about zooming in, focusing on the individual components, the organs, tissues, and cells, to gain a deeper understanding of their roles and functions. Imagine a complex machine, say a car. To know how it works, we would need to understand each part, how the engine functions, how the wheels turn, how the brakes stop the vehicle. Similarly, in biosynthetic studies, understanding the whole organism requires a deep dive into the individual components. Let's take isolated organs, for instance. By studying an organ separate from the body, we can gain insights into its specific functions, independent of other influencing factors. The heart, in isolation, can help us understand the dynamics of blood circulation, or the liver can shed light on detoxification processes. Similarly, isolated tissues and cells offer a rich field for exploration. By isolating specific tissues, we can observe their unique characteristics and roles. For example, studying muscle tissue can help us understand how movement and strength are generated. On a smaller scale, studying individual cells can unravel the secrets of life at its most basic level. With each cell being a microcosm of life, understanding its workings can provide a blueprint for comprehending more complex, multicellular organisms. This approach of reductionism has revolutionized biosynthetic studies. It has enabled us to piece together the complex jigsaw puzzle of life, one piece at a time. And while it's true that the whole is often greater than the sum of its parts, it's equally true that a fuller understanding of the parts can lead to a greater appreciation of the whole. Studying life piece by piece, we can construct a fuller picture of the whole. With each discovery, each insight, we move closer to unlocking the mysteries of life itself. Biosynthetic studies aren't just about understanding the normal, they're also about exploring the abnormal. The tools we wield in this exploration are as fascinating as the discoveries they lead to. Two such tools are grafts and mutant strains. First, let's talk about grafts. In the world of biosynthesis, grafting is a surgical operation that joins two living organisms into one. It's like creating a biological chimera, a creature of myth, but in a laboratory setting. This technique allows us to observe the interaction between different genetic makeups within a single organism. Imagine an apple tree. Now, graft a branch of a pear tree onto it. The pear branch continues to bear pears, even though it's now part of an apple tree. This is because the genetic information for pear production resides within the cells of the pear branch. This experiment gives us a real-world demonstration of how genetic information controls biosynthetic processes. Next, we have mutant strains. Mutations, in the simplest terms, are changes that occur in an organism's DNA. These changes can affect the biosynthetic processes in an organism, leading to variations in physical traits or biochemical functions. By studying these mutant strains, we can understand how different genes contribute to biosynthesis. Imagine a field of corn. Each stalk looks similar, but there might be one that's shorter or has a different color. That's a mutant strain. By comparing this strain to the others, we can identify the genes responsible for height or color in corn. Understanding these variations is not just about satisfying our curiosity. It's about unlocking the potential of life itself. Through these tools of grafts and mutant strains, we get a glimpse into the vast spectrum of possibilities that exist within the genetic code. By studying the abnormal, we learn to appreciate the normal. And by understanding the normal, we can begin to dream of the extraordinary. Through understanding variation, we gain insights into the vast potential of life. So, what have we learned about the methods in biosynthetic studies? We've delved deep into the world of biosynthetic studies, unraveling the intricacies of life's building blocks. We've seen how trace techniques enable us to follow the breadcrumbs of biological pathways, helping us understand where and how these pathways intersect and the role they play in the grand scheme of life. By tagging molecules with radioactive or stable isotopes, we can track their journey, revealing the secrets of biosynthesis. We've explored the power of reductionism, breaking complex systems down to isolated organs, tissues, and cells. This approach allows us to create controlled environments, minimizing variables and enabling us to delve deeper into specific processes. It's like taking out individual pieces from a puzzle to examine them closely, then putting them back together to see the bigger picture. We've seen how grafts and mutant strains provide invaluable tools for understanding variation. By creating chimeras or inducing controlled mutations, we can observe how these changes ripple through the biosynthetic processes, affecting everything from individual cells to entire organisms. This not only offers insights into the mechanisms of biosynthesis, but also helps us understand the factors that drive variation and evolution. But what does this all mean? Why does it matter? Well, these studies are not just about understanding how life is built. They're about understanding how life works, how it adapts, and how it evolves. It's about seeking answers to some of the most fundamental questions of biology, and by extension, of our own existence. So, as we conclude, remember that this is just the tip of the iceberg. There's a whole world of biosynthetic studies waiting to be explored. And each method, each technique, each tool we've discussed today is a stepping stone on this journey of discovery. Subscribe for more because you won't find me later. Stay curious, stay informed, and continue exploring the wonders of life.